Across the world, over 50 million people suffer from a medical condition that is too often misunderstood. Some people believe that it is contagious, or that those who have it are mad and should be hidden away, or even that they are possessed by evil spirits. But epilepsy is none of these things. So what is epilepsy? Epilepsy is a chronic disorder of the brain characterized by recurrent seizures. These are brief disturbances in the electrical functions of the brain. People with epilepsy, they often struggle with anxiety and depression. Because of the stigma they face, it can be difficult for them to live a full and normal life. 70% of people with epilepsy can be seizure-free with adequate treatment.
It is estimated that more than 12 million people in India have epilepsy.
And over 70% of these do not have access to proper treatment.
Rapid urbanization, social stigma, limited knowledge of doctors on epilepsy are all contributing to increasing the treatment gap.
The most challenging component of persons with epilepsy are those 30% who do not respond to medical treatment. These patients are said to have drug-resistant epilepsy. Such patients continue to have uncontrolled epilepsy, day after day, month after month, and year after year. No treatment is effective to control or cure their epilepsy, and most of them develop severe intellectual disabilities. Their risk of death from epilepsy is also very high, and most succumb to their illness after a prolonged miserable life. These patients suffer from high degree of social discrimination, and are ostracized from the society due to stigma. There is little that is known about the mechanism of drug-resistant epilepsy. However, a lot of recent studies have shown presence of large abnormal networks in the brain, which repeatedly generate abnormal amounts of electricity, leading to uncontrolled seizures. These networks are dysfunctional at a molecular and cellular level, making them resistant to any form of medical therapy. Surgery is the only option for these patients. However, the surgical strategies are complex and require extensive brain mapping techniques prior to surgery. High-end investigations like video electroencephalography, high-resolution MRI, ictal single-photon emission tomography, positron emission tomography are required. Surgery performed in an experienced center can yield excellent results. However, despite best of available investigations, up to 40% patients may not qualify to become eligible candidates for surgery. Thus, it is important to have collaboration with high-end research laboratories that have advanced molecular and intracellular electrophysiology techniques that can provide better understanding on the mechanisms of epileptogenesis.
Understanding the immense need to develop this, the Department of Biotechnology, India provided a generous research grant to develop a center of excellence for epilepsy. This was developed as a collaborative project between the Premier Institute, the All India Institute of Medical Sciences, New Delhi, and National Brain Research Centre, Manesar. The collaborative project was approved in the year 2011.
The Center of Excellence for Epilepsy was established with an objective to find a cure for epilepsy, by understanding mechanisms of epileptogenesis by studying epileptogenic networks, potential biomarkers. The same is aimed to be achieved through strategies utilizing translational methods from bed surgery laboratory approach. There are 4 key verticals in this project. This is for the first time that such a project has been conceived for India, and also for the entire Southeast Asia. The first vertical is establishing an advanced brain mapping facility consisting of magnetoencephalography at the National Brain Research Center. Magnetoencephalography is a facility which records brain waves by recording the magnetic signals emitted by the neurons. These signals are very minute, about 1 million times less than the Earth's magnetic field. To prevent the interference from the Earth's magnetic field, the whole machine is encased within a room, which is magnetically shielded. It was also important to place this machine where the ambient magnetic noise generated by traffic and electricity is minimal. Considering this, the National Brain Research Center was tested to be an ideal location to place this machine. The magnetoencephalography studies brain activity by recording magnetic fields produced by electrical currents occurring naturally and evoked in the brain using very sensitive magnetometers and gradiometers, coupled with superconducting quantum interference devices maintained at approximately -269 degrees centigrade. The magnetic signals are in the order of 10 to the power -9 to 10 to the power -15 Tesla. A separate building was constructed to house the magnetoencephalography machine. The building has a separate reception, a 10-bedded patient ward, MEG preparation lab, MEG research lab, and the MEG room itself. The Magnetoencephalography Center was inaugurated by the Honorable Minister for Science and Technology, Dr. Harsh Vardhan. This was in presence of Professor P.N. Tandon, President NBRC, Professor Subrat Sinha, then Director of NBRC, Professor Manju Sharma, and Professor Vijay Raghavan, then Secretary of Department of Biotechnology. Also present were Dr. Manjari Tripathi, Professor Neurology and in charge of the MEG Center from the All India Institute of Medical Sciences, New Delhi, and Dr. P. Sarat Chandra, Professor, Neurosurgery and Team Leader of the project, from the All India Institute of Medical Sciences, New Delhi. The magnetoencephalography at the National Brain Research Center is first of its kind of installation for the whole country. There are only 2 such installations in India now. The MEG Center at NBRC serves as a referral center for the whole north, east, and west of India. We will now show you a short clip showing how the procedure is performed. Once the patient comes to the center after the scheduled appointment, a detailed history is taken regarding epilepsy and the medications the patient is on for epilepsy. The patient and the attendants are also explained the detailed nature of the procedure. A detailed screening is performed to ensure that there are no metallic objects within the vicinity of the patient, which may interfere with the recordings. The patient is then taken into the MEG preparation lab. And 128-channel high-density EEG cap is then placed over the patient. This for performing simultaneous EEG and MEG signals. For co-registration of the MEG signals over magnetoencephalographic images, digitization is performed. The whole preparation is a painstaking procedure and requires about 1 to 1.5 hours. A high degree of skill and training is required to perform this. The patient is then shifted to the MEG machine, located within the magnetically shielded room. MEG is a completely non-invasive procedure and takes about 2 to 3 hours. The MEG signals generated are then processed, and using advanced software is then superimposed on the 3D images of the brain. Magnetoencephalography provides critical information about the location and generation of epileptic signals in the brain. This is not only useful for surgical strategies, but also provides very useful information regarding the mechanisms of origin and spread of epilepsy in the brain. Over 3,000 MEGs have been performed till date, not only from all parts of the country but also from countries like Dubai, Nepal, Kenya, Muscat, Pakistan, and Bangladesh. The second component of the project is the Epilepsy Neurobiology Lab. This is located in the Convergence Center at the All India Institute of Medical Sciences at New Delhi. The lab again first of its kind for whole of Asia and has advanced facilities for intracellular electrophysiology. The lab was inaugurated by Professor P.N. Tandon, and is named after him, acknowledging his contributions towards the development of neurosciences in India. The Neurobiology of Epilepsy Lab is equipped with high research instruments, including facilities to record electricity from single neurons. The research lab is unique in the sense that it studies functioning neurons from freshly resected surgical sample, made available to it within minutes after surgery. This provides very important information regarding the cellular mechanisms of epilepsy and its generation. The Advanced Molecular Lab is located at ACBR, New Delhi, which allows study of molecular basis for mechanisms of epilepsy.
The clinical collaboration is being done with the Department of Neurology and Neurosurgery at the All India Institute of Medical Sciences, New Delhi.
Epilepsy surgery is a multi-team approach and is supported by departments of neuroradiology, nuclear medicine, and neuropsychology. Complex multimodal imaging is required to understand the origin of the epilepsy in brain. Epilepsy protocol thin slice MRI, MRI tractography, PET MRI fusion are some of the sequences required to map the brain. In addition, intraoperative electrocorticography neuromonitoring is also available. The Department of Neurology is equipped with state-of-art video EEG stations. The Department of Neurosurgery has all the modern facilities, like high-end operating microscopes, intraoperative neuromonitoring, neuronavigation, intraoperative MRI, robotic systems, stereoencephalography, to perform all complexities of epilepsy surgeries.
The center has published more than 300 scientific peer-reviewed papers in high-impact factor journals. These include some of the world's highly rated journals like the New England Journal of Medicine. The team have established the safety, efficacy and feasibility of performing highly complex epilepsy surgeries earlier not performed in India. They have also described several new surgical techniques now acceptable all over the globe. Over 100 papers have been published in the areas of pure basic neurosciences, which have shed new light in the mechanisms of epilepsy at the cellular and molecular level. All these have brought us closer to understand this complex and difficult disease.
The Epilepsy Neurobiology Lab is focused on identifying biomarkers for defining the epileptogenic zone, as well as biomarkers for drug-resistant epilepsy, using multi-omics approaches, like genomics, proteomics and lipidomics. In addition, we are evaluating the therapeutic potential of identified molecules by conducting mechanistic functional studies on patients as well as animal model of drug-resistant epilepsy. We demonstrated for the first time, reduced endogenous kinesynthesis contributes to enhanced glutamatergic activity in hippocampal sclerosis. This was due to altered levels of CAT2 enzyme and its cofactor PLP. This study provides a strong evidence that application of KINA may have therapeutic potential. Our group reported for the first time the RNA sequencing analysis of surgically resected tissue specimens from hippocampal sclerosis patients. We demonstrated the first direct evidence that large-scale networks exist at the cellular level in patients with hippocampal sclerosis and demonstrated two aberrant networks, one emanating from the hippocampus, and the other from the anterior temporal lobe.
Our work on liquid chromatography and tandem mass spectrometry identified altered lipids in resected brain specimens of cortical dysplasia. Our results suggest that increased TAG levels could serve as potential biomarker. Specific lipid mass spectra could thus be used for defining the epileptogenic boundaries, for precise resection during epilepsy surgery, using intelligent iKnife approach. The publication in the New England Journal of Medicine was considered among the top 10 papers for the journal, for the year 2018. This article now has more than 300 citations, and is now a landmark paper. This is for the first time that such a study from India received international acclaim and attention. The center has trained more than 10 PhD students, 6 fellows, several visiting students and has conducted more than 20 workshops and conferences. It has also helped several places in the country to start an epilepsy surgery program. In addition, it has spearheaded to start epilepsy surgery programs in other countries like Indonesia and Morocco. Epilepsy is a disease not to be feared. It is to be understood. It is a disease to be treated, not be stigmatized. 1 person out of every 100 people has epilepsy. Over 70% respond to medical therapy, but over 30% do not respond to any medications. Such patients need to be worked for the possibility of surgery. Surgery performed at a tertiary center is highly effective. There is also an urgent need to develop a strong research arm. We will not be away from the truth, stating that India requires a national epilepsy center, where all the facilities may be integrated and available under a single roof. Such efforts will go a long way to understand, treat and finally cure this terrible malady. The MEG Resource Facility and the Center of Excellence for Epilepsy have made a sincere effort in that direction. It takes time to build such national assets, the most important being training, skill development, and a push to encourage more doctors and scientists to take this up as their career.